Okay, thank you very much. So this is my first ICG, so I'm new, and I will explain a little bit later why that is. But I first want to take you a little bit into my world. So as Shane said, my background is neuroscience. I'm interested in trying to understand the brain, and kind of questions that keep me busy are questions very basic, like how many cells do we have in the brain? But also things like how many types of cells do we find in the brain? And of course, that's very important. I mean, how do you define a cell type? How many proteins are expressed in the brain? What cells express those proteins? Where are those proteins distributed? With what other proteins are they interacting? What's their function on a cellular level? And then also going into diseases, trying to identify how diseases, how they occur, and also what the molecular components of different pathological processes are. And these, of course, are very, very complex questions that we cannot answer with a single essay. So what we need to do is we have to somehow increase our completeness. And I think what we need to do is almost like zoom in and zoom out. We need to, I mean, especially for brain, we know that it's a very complex organ, so we have to sample many regions. We had the same, I mean, when we talked this morning about the microbiome, is that also we have to maybe sample different parts of the gut in order to be able to get a complete answer. So these are the things that we have to do, and I think doing science in this era is one of the times where I think we are getting close to be able to zoom in and zoom out. So also I want to say a little bit about, this is kind of technical from neuroscience, so working with brain is a little bit, I mean, we have special challenges. Some of them might also be familiar with people working on other types of samples, but we have main cell types, which we all know, but also we know that there's many subtypes. We also know that not all cell types are in the same size. The population sizes are very different. There are some really rare cells, and there are some cells which are occupying a large proportion of cells. There's different brain regions which have different cells, but also things like extracting cells from brain. It's impossible. Maybe you can extract microglia cells from brain, but most cells, they don't like to be extracted, which means that we are limited to nuclear. If we want to do single cell, we might have to go for a single nuclei. And then things like the transcription bursting is even more of a kind of problem, because these are very temporary events, and we only measure what happens in the nucleus. So these are all the kind of challenges that we work with, and we have to kind of find ways to be able to answer our questions. So then when I kind of see, let's at the level of transcriptomics. So as I said, so I'm working in the Human Protein Atlas. So we are mainly focused on proteins, but of course also protein expression, and therefore we are interested in transcriptomics. And then you can, as I see, there's kind of three flavors. So there's the bulk transcriptomics, so where you take a sample and you sequence all the RNA molecules, which is a combination of all cells. And of course that gives you possibilities to sequence really deep. And I think you can sequence as deep as detecting all genes or close to all genes. But also, of course, you have some dilution problems there because you don't know where your transcripts come from. And if there's really rare cells, it might be that they get diluted out by other transcripts expressed by cells which are more abundant. Then when you go to single cell, then of course the single nuclei is the only option almost. And then of course there's all kinds of technical problems. Not only this has to do with the fact that there's only single nuclei, but also the fact that some cells somehow don't like to be isolated. So you have to maybe do some FACS sorting approaches to be able to kind of isolate those cells. And it's really difficult to recompose the tissue and kind of put all the data together to kind of see what your bulk transcriptomics would be like. So then the spatial transcriptomic and the stereo-seq is, of course, the next promise. Of course, we have to see if it kind of holds. But there you would, in theory, be able to compile all your data back to a bulk sample, but you would have the resolution to look at the individual cells. And I think that's a promise, and it kind of makes me really kind of jump up my chair when I kind of read about the possibilities. The other thing is, of course, we want to look at proteins, and I think proteins is the kind of, these are the functional units. These are the units that are giving the function, where the transcripts are only the kind of prediction that those proteins are actually there. And then you can look at things. So we are in Human Protein Atlas mainly working with affinity proteomics using antibodies. And then, of course, you can go to immunohistochemistry, looking at protein distribution. You can use immunofluorescent approaches where you have the additional benefits of being able to multiplex. But also you can kind of go from three-dimensional kind of sections going into three-dimensional. So being able to look into an entire three-dimensional tissue piece to look at where proteins are located and how cells or proteins are interacting. So, and that's a little bit of the kind of the questions and the toolboxes that I'm using, or that we are using. And this is a little bit of kind of our timeline. So the Human Protein Atlas project started in 2003, and in the beginning, it really had an antibody focus. It really had a focus on generating antibodies against every human gene, being able to visualize the location of those proteins in human tissues. And that was completed in around 2015. And that was also the time that the next generation sequencing started to kind of become accessible to us and to many, and we started to use it first as a kind of validation to see, do we see our antibody bind where we have the transcripts? That was kind of how we started working with transcriptomics. But then, of course, we also very quickly discovered the kind of the added value of having transcriptomics data. So, and that's one of the things we did around the time. And also then we started to explore more and more tissues. So we integrate lots of data from the Broad Institute, from Phantom, to really be able to see, look at more brain regions, being able to look at the protein expression there, also adding more species, and also lately we kind of did an experiment together with MGI, so using the T7 sequencer, which was at Karolinska, to analyze about 1,000 samples covering 190 regions of the human brain, to be able to kind of zoom out, to be able to kind of get a better coverage of gene expression in the human brain. And recently, since the beginning of this year, we also started exploring stereo-seq, so being able to use this kind of or the subcellular resolution, being able to take a whole piece of a human brain and be able to analyze the gene expression. So I will show a little bit more about that later. So the current version of the Human Protein Atlas, there's a focus, and Matthias Ullén will talk about the Human Protein Atlas in the precision medicine session tomorrow. But we basically organize our data when it comes to organs, tissues, cells, but also things like metabolism, diseases. So you can find all kinds of information about proteins, and you can, and we try to have it in a really integrated way, so that the data can be integrated. The brain session, or the brain section as it is at the moment, so we really try to provide an overview of gene expression, or protein expression, I would say, in all major regions of the brain. So you will find a lot of data. So this is, for instance, when we sequence our 1,000 samples of different parts of the brain, you can really see that based on the gene expression, they cluster quite nicely. You can see that cerebellar cortex is a kind of isolated group. You can see all the cortical regions and the forebrain regions clustering together, and then you kind of go from the forebrain all the way up to cerebellum. You can see a very nice kind of profile of gene expression. So the data looks quite nice, and you can find all the kind of marker genes for different brain regions quite nicely organized. And this can be a nice resource for trying to find genes which are differentially expressed between different parts of the brain, but not single cells. Also, one of the things we did in collaboration with MGI, BGI as well, is to look, for instance, at other species. So look at the pig, look at the mouse, and also give the transcriptomics profiles of those ones. We're also working on rat, we're also working on macaque. So we want to kind of extend, to get a little bit of an overview of the mammalian brain, allowing you to look at differences. Also, we have a lot of immunofluorescence data on the mouse brain. So the mouse brain has the big advantage of being small, and it's quite easy to sample all possible areas and cell types in only a few microscope slides. But also we have data on human brain areas using the tissue microarray approach, where we have small punches and we can look at protein distribution at the cellular or subcellular level. So then a little bit about what we have learned so far, or what we have learned. So one of the things is that we published 2 years ago, is that we started to combine all the data from and try to look at different parts of the brain and see if there's some kind of mammalian architecture. Is there some kind of blueprint of the mammalian brain? And we could find it quite well. That many genes you find in the same region, or in the mouse or the human or the pig. You can see that, for instance, in the mouse and the pig, the olfactory bulb is kind of developed in a way. I mean, smell is quite important compared to humans, so there you see some variation. But in general, you could say that there's quite some similarity. But there was also some differences, some clear differences. And for instance, here we have a protein which is called secretagoguin, which is very highly expressed in the human cerebellum, but you don't see it in the mouse and in the pig. So there you see that there's a gene which is clearly different. We also looked a little bit at transcription factors and transcriptional regulation, and you see that if you look, so here we have the 3 different species, that if you look at different types of transcription factors, you see that they are kind of distributed in the same way. There's some exceptions, for instance, here the olfactory bulbs, you can see some variation, but in general, I would say the transcription factors are distributed equally. But what's interesting is that if you look at many functional proteins, and especially receptors, we find quite a lot of variation between species. And some are now, for instance, opioid receptors. So the mu-opioid receptor is quite highly expressed in the human cerebellum, which is not expressed in the mouse or the pig cerebellum. And if you look at the literature, there's very little known about the function of the opioid receptor in the human cerebellum or in the cerebellum, because there's no model systems that can be used to test this. To illustrate it even further, another receptor is the nicotinic receptor, and I don't know if any of you had neuroscience courses when you were studying, but the kind of textbook knowledge is that you have 2 different forms of the brain nicotinic receptor, which is the alpha-beta subunit or the alpha-7 heteromeric subunit composition. And then if you look at different comparisons between human, pig, and mouse, then if you look at the distribution of the different isoforms at the level of the tissue, you see that there is some variation, which probably indicates that this textbook knowledge is not the complete and the full truth, but there are some other variations. Like, for instance, in human, we see quite a lot of alpha-2. We see that in the pig, especially here, we even have the epsilon subunit in the basal ganglia, which we didn't see in the other species. So it seems that the kind of textbook knowledge is mainly based on the mouse data, and it fits quite nicely to the mouse data, but it seems that in mammalian biology there's more things happening. Another thing is that we really started to collect samples with a very good anatomical description. So sometimes when you read papers, they compared liver and brain, and I always wonder what part of the brain did they take. So here we used micro punches, very small punches, and we could look at the prefrontal cortex, which is a part of the brain which is really developed in humans. And by having these really detailed samples, we could compare them, and we could look at their landscape, and we could already identify very small subtle changes. And for instance, what we found is that many of the neuropeptides and neuropeptide receptors are differently distributed in different parts of the prefrontal cortex. And we found that this kind of more medial and more ventral part seems to be a region which has more neuropeptides and more neuropeptide receptors, which was quite interesting. So we were able to get to the detail or to the level where we could compare closely related regions of the brain and try to find some interesting changes. So now I will go into, so this was kind of zooming in, now I will zoom out again, because sometimes to understand biology, you need to have the full overview, and you need to have almost a 3D overview. So this is a study where we looked at the locus coeruleus, which is a part in the brainstem, which seems to have the first signs of Alzheimer's pathology. And questions are like, are all cells in this brain part, do they have the same sensitivity? Do we see some patterns? Is there a part of the locus coeruleus which maybe gets tangles first, or is it more randomly? Is there some random spreading, or is there some kind of spreading between cells, or are some cells which are in the same area more vulnerable? So for this, we used whole tissue blocks and used some iDisco method and using some clearing methods to be able to reconstruct a full three-dimensional part of the brain. And things that we found there is that you can look and you can identify different tangle-bearing cells inside the locus coeruleus, and you can really, in a three-dimensional space, reconstruct how far they are apart. And is there a random distribution, or do you see that sometimes cells come in pairs or in mini-groups? And that's one of the things that we saw, is that within the locus coeruleus, we saw that the kind of dorsal part seems to be getting tangles before the ventral parts. But also we saw that quite many of those cells, they are kind of in small mini-groups. And you can do all kinds of statistical analysis to see if it's random or it's more kind of leading to or looking as spreading. And we found that there's some enrichment. So if a cell has a tangle, there's a higher probability that a neighboring cell also gets a tangle. And then I will move into the thing that we started now. As I said, it was really at the end of my timeline. So this is really something that my lab is working on now a lot and doing experiments on a weekly basis, trying to use this method to understand how the molecular and cellular organization of the nervous system is. So here we have a spinal cord. So this is a project where we're looking at ALS. And this is the section blocks that we received. And now we are doing. So I'm not giving all the parameters of how we do it. This is kind of the standard stereo-seq protocol as we use it now. And we get this nice overview. So here we can do some basic single-cell-based clustering analysis, try to use different size of bins. So basically, you can either combine 100 by 100 spots or 50 by 50 spots. And then you can ask the question is, how similar are the spots to each other? And then you get kind of plots like this. So you can see that there's different clusters. If you make the bin sizes smaller, you can see that even some of those clusters are kind of isolated, which means that they could be subcells or cell types. And then you can, of course, project those back onto the tissue and see where your different cell types are. And here you can see, as a kind of control, you can see that there's a lot of white matter and a lot of gray matter genes, which you can find nicely organized. You can see that there's a border around the tissue, which we can see. So in a way, we are getting a lot of information about the organization, or at least the molecular organization of our tissues. And these are a few other examples. What's quite nice is that also, for instance, here you can see that even if you look at the distribution of GFAP, an astrocyte marker, you can almost see blood vessels, and you can get a lot of information about the distribution of those proteins. We did the same thing on human cortex. And here, this was kind of one of the first experiments we did, so we went a little bit further. And you can see that if you use a kind of large bin size, you can very nicely identify different layers of the cortex. We know the cortex has 6 layers, which we can nicely identify by looking at the clustering. We can very nicely see a difference between the gray matter and the white matter. And then when you go down into the size of the bins, all the way up to bin 20, which is about 10 by 10 micrometers, you can already start seeing clustering on smaller sets, which could be individual cells or parts of cells. And of course, then what we would like to achieve is true single cell. So we have the resolution of single cell. But we would like to know where does a cell start and where does a cell stop, and which mRNA or which spots belong to which cell. So if you zoom into a small part of the gray matter from this sample, and you just kind of plot your UMI counts, I think what you can see and what you can appreciate is that you see that there might be individual cells. So those spots where the UMI counts are a little bit higher might be spots where there would be a single cell. And try to find ways of creating some kind of outlining. So a postdoc in my lab, Emma Gerrits, so she's trying different approaches to look at, can we use that neighboring UMI counts? Can we somehow create some kind of hotspots or some kind of concentrated spots to be able to draw where we think a border of a cell could be? So we can use all this kind of density-based clustering. We can look at finding the centers of those clusters, and then kind of try to create something like this, which would be a theoretical map of all the different cell types, which would then allow us to put all the spots which are underneath those masks back into the cell, and be able to then say something about single cell. So this is kind of where we are at the moment. So we are, as I said, doing a lot of experiments, trying to get as deep as we can with our sequencing, try to get as much information out of our material, and then, of course, aiming to get to single cell resolution. So a little bit as a summary. So we kind of provide our data on the atlas, so we're sharing our data. We really would like to integrate lots of different omics. I mean, at the end, we would really like to do it in a single experiment, but I guess that's something that's far, far ahead of us. We are kind of increasing our coverage. We're kind of trying to zoom out and zoom in to be able to get more information about protein distribution in the brain. And I guess we're using 2D methods, 3D methods, but I think the main thing is that we, with StereoSeq, we really feel that we are getting to the single cell resolution, but I think the main question is, can we make it as true single cell resolution? Because I think that would be a big game changer for the field. I would like to thank many people. So many people in the Human Protein Atlas, which is a huge project involving hundreds of people. My group at Karolinska Institute, the HPA Brain Group. Then collaborators of BGI, MGI, which are involved in setting up the method. But also, especially Professor Palkovic in Hungary, who has this micro-punch method and has punched out very small parts of the brain at the Semmelweis Brain Tissue Bank. Also the Netherlands Brain Bank for providing lots of tissues. And some of the funders, which especially the Knut and Alice Wallenberg Foundation, which funded the Human Protein Atlas over the years. So thank you very much, and I'll be happy to answer any questions.
Thank you, Jan, for the great talk. And is there any question from the audience? Yes, please. Tim?
For a great talk, really. Maybe for the last with the StereoSeq use, and you use the UMI counts. Have you looked at more dense regions like immune infiltrations, in the sense if you can use the same technology, or could you segment or deconvolve a spot when there's many different signatures?
I mean, one of the issues we have is that I think the spots that we detect are the neurons. So neurons are, I think they are in size, they are big. They have a lot of mitochondrial genes, so they're high in mRNA counts. I think also one of the problems is that many of the other cell types, they have very low mRNA counts. There might be a factor 10 between them. Also in our section, it might even be that 10 micrometer might be too thick for some cells, which are even thinner than that. So you might even have two cells which are on top of each other. So I think there's all kinds of challenges that we have to kind of found. I mean, the thing is also that we know for many cell types we have a kind of a list of our marker genes or signature genes. So also one thing is to be able to use those signature genes to identify spots where there would be a high probability that there would be a certain cell type located. So we're trying all kinds of approaches in that direction.
Perfect. Yeah, thank you.
Yes, please, Willie.
Thank you so much for your informative presentation. One naive question, actually. So you have been talking about showing good StereoSeq and a single-cell gene expression profile on single-cell level. I'm wondering if you have done or doing the similar study on proteomics to figure out the resolution, the similar resolution on proteomics, not necessarily to be entire gene, but in case you found differentially expressed gene down or up regulation, do you also double check on protein level to see if there's any correlation?
Mm-hmm. Yeah, so I mean, proteins is like, I mean, if you look at I mean, we have done several studies within the Human Protein Atlas to see is that how can you predict protein concentration based on mRNA concentration, and that's something that's not there's no I would say factor which would say that if you have this amount of mRNA you would have that protein. It seems to be that for each gene, for each protein, the concentration will tell the RNA concentration will tell the protein concentration, but it will vary for all different proteins. So then the other thing is that, which is kind of difficult, is that for brain, the proteins might be in other parts of the brain. So sometimes we have neurons, let's say in the back of our brain, with axons going all the way to the front of the brain. The proteins will be here, the mRNA will be there. So how are you going to correlate, or how are you going to correct for these things? So it's quite complicated task to do. Yeah.
Yes. Question.
Yeah, you just showed the bin 20, so 10 micron brain sections, and I saw this was more than 56 clusters. So when you compare this with a single cell of the same kind of section, what is the relation in numbers of single subtypes? So
I would say the first thing is that we would not be able to do single cell, but single nucleus. So which would already kind of lower, I think, our. And I think you can, if you include enough cells, you can decide your kind of how many clusters you want to detect, and you can have subclusters. I think the thing that we should maybe aim for is not the number of clusters, but how isolated those clusters are. So are you able to identify really isolated clusters? Or is it a big transition going from one cluster into the other cluster, and you just, by mathematical measures, you kind of identify subclusters, but it kind of is one continuum. So I think which we should aim for, having those isolated clusters, because I think that's when we get to single cell type of resolution.
But it was not all these 56 were isolated, right?
No.